Okay, so last time we talked, we met about the 5 different categories of the temperatures that the organisms like. What was the coldest? Does anyone remember? Well, the name, the general name. What was it? Psychrophiles. Psychrophiles, meaning cold loving. And then these guys were next, psychrotrophs. What were the ones that were the middle lovers? Mesophiles. Excellent. Remember M for middle. How about the next group? Thermophiles. Excellent. And then our last ones? Hyperthermophiles. Exactly. They like it hot. All right. Hot, like boiling. They like it like that. Okay. So one of the things to keep in mind and why this is important to know temperatures for bacteria, and we haven't covered this slide yet, right? No. Okay, good. Is that this has to do with how we store our food. We talked last time about storing eggs in the refrigerator here because we clean them and when you take the bloom off. The same is true with other foods. We have kind of a danger zone that we're aware of, where if we put our food at around a 60 to 120, we get a really rapid growth of bacteria. The reason for that is in that range, mesophiles and thermophiles can grow. So we're getting 2 different groupings. And then also these are organisms that tend to make endospores. And in this group is an endospore former that we all fear, which is one that's called Clostridium botulinum. You guys have probably heard of it before as botulism. And so this is an organism that we all live in fear of because normally our food is cooked carefully, those spores aren't present, and we're safe. But if we heat our food, let's say we're at home and we heat our food, and then we leave it sitting out on our countertop for a day or 2, any spores that were there that didn't get destroyed through the heating process are now going to be growing into vegetative cells. This is why when you cook food, like let's say you make a meatloaf at home, right? We're heating that meatloaf, but we're not heating it enough to kill endospores. And then if you let it sit out for a day or 2, it's just going to have a ton of vegetative growth. So we want you to make sure you refrigerate things carefully. So that's why we refer to that as like a danger zone. Okay, quick check. A psychrophile grows best at what temperature? I heard it, D. Excellent. Yeah, somebody's been studying. She's like, D. So 15 degrees Celsius. Yes. How many general categories exist for microorganisms based on their temperatures? 5. 5. Remember, psychrophiles, psychrotrophs, mesophiles, thermophiles, hyperthermophiles. So those are our 5.
So we only need to learn the highest you could go, right?
No, you want to learn the optimum. So what its preferred is. Yeah, because you get too much overlap. If you're trying to learn the lowest and the highest, it gets confusing because sometimes you'll have similar numbers. I just want you to know their optimums, their favorites. Now, the other thing that affects bacteria besides temperature is things like pH. So bacteria are like humans. They prefer to have a neutral pH. So we like to have a pH of around 7 in our bodies. Sometimes our blood gets a little acidic because of CO2, so we run between 6.5 to 7. And that's where most bacteria like to grow as well. And that makes sense because a lot of our mesophiles that live with us would want to be used to our body conditions. There are a few bacteria out there, though, that are referred to as acidophiles. And what does that sound like? If they're an acidophile, what does that sound like? Yeah, they're acid lovers. They like the acidic spots. So these are going to be ones that grow in acidic environments. Where do we have an acidic environment?
Our stomachs.
Our stomachs, yeah. Have you ever heard of ulcers? Yes, ulcers are caused by an acidophile. It's a bacterium that can grow inside of our stomach. And one of the things we used to think caused ulcers was stress. Well, it turns out they go hand in hand. When you get stressed, your immune system drops. And when your immune system drops, it allows this bacteria a chance to start to grow. Because your immune system's being affected. Your stomach acid levels are affected when you're stressed. Everything's affected when you're stressed. And I can tell that here every year because of during finals week. When everybody comes in for finals week, everybody's coughing, sneezing, sick. It's like one of those things where we almost always get sick during finals week or sick during midterms. And we're like, well, why does that always happen? It's such bad timing. It's because we're stressed. So anytime you have something stressful going on, your immune system plummets. So another thing that can affect them also is osmotic pressure. So remember, this has to do with how much salt or sugar or pure water is in an environment. So, for example, a hypertonic solution, something like seawater, can cause plasmolysis. That means that the water will leave the cell and it will cause the cell to break down. Okay, cells don't like to be shriveled up. Now, we use this to our advantage. A long time ago, our ancestors learned that they could preserve meat and foods by doing something to them. Does anybody know what they used to do to them? Add salt. Yeah, they'd salt the heck out of them. We actually still do this in certain places where we don't have a lot of electricity. We do it a lot like in Mexico. We do it a lot in places of Africa. We do it a lot in different areas where if electricity is not always dependable, or maybe you're just out in the wild area, you can do a lot of salt and that preserves your food. And what this salt does is it pulls the moisture out of the bacteria cells. It causes them to shrivel up and then they die. We see this ourselves. If you're ever eating salty popcorn, let's say you go to a theater and you eat salty popcorn, your lips start to feel a little dry and chapped. You need a little bit of that big Coke. You know, they give you the bucket of Coke or whatever you buy. I always am shocked at the sizes there, right? They give you like the bucket and you're like, okay, yeah.
It's like $30.
Yeah, the $80 bucket of Coke. Yes, exactly. And we need that to replace the water that we're losing with that salty popcorn. They're smart. They got us figured out. Now, we mentioned in our last lab that some organisms are able to handle the salt. Remember, which genus did we say can handle salt? Excellent. Staphylococcus. Excellent. So Staphylococcus can handle salt because it's able to adjust pumps in its plasma membrane that if it's put in a salty environment, it quickly pumps salt inside of itself. That's brilliant. It balances itself out. It matches its environment. So if the environment's salty, it'll make itself salty. Okay? So we refer to those guys as a facultative halophile. So facultative means they can turn it on or off. Okay, so on or off. We also have some halophiles that are obligate halophiles. And what this means is they can't turn it off. So this is why I can't put seawater organisms in fresh water. Because their pumps still pull salt inside of them. They have no control. And it causes them the opposite problem. They swell up and burst. We have a halophile that is an archaea that's very important to us. And it is one that gives flamingos their pink coloration. Have we talked about that before? No? So there's a certain little archaea that lives in the ocean. We'll talk about them a little bit later. And it has a natural pink pigment to it. And so shrimp that are filter feeders, that's their favorite food. They eat this pink archaea, and guess what color shrimp are? They're pink. Yeah. And crabs will eat it. That's why they're kind of red. They like the same filter feeding organism. And it turns out flamingos turn pink because they eat the shrimp and the crabs that ate the archaea. This is why when flamingos are born, if you ever see baby flamingos, what color are they?
White.
White. Yeah, when they're first born, they're like snow white. And you're kind of looking at them going, that's not a flamingo. That's a white bird. I don't know what that is. And it turns out they have to eat enough of the shrimp and the crustaceans to actually get their pink pigmentation. We learned this the hard way. The first time we put flamingos on display at a zoo in the early 1900s, they captured them all in Florida, they put them in a zoo, and they all turned white within a few months. And the zoo was like, we don't know what's wrong with them. It took a while to figure out they needed proper food, that they needed shrimp and crabs. Do you have a question?
In the desert, in Argentina, there's like a bird that is like salt and they're red.
Ooh, so they're probably eating the same type of archaea.
Yeah.
I love that. I love that. And if you ever go to places like the Red Sea, what makes the Red Sea red? The archaea. Isn't this cool? It's like cool stuff, right? Okay, so here's a picture of what's happening in these cells. Again, if I put a cell inside of salt water, it's going to lose its water. hypertonic, E for exit. The water is going to exit, and it's going to shrivel up, go through plasmolysis, and it's going to die. Again, the way our halophiles get around this is they have little pumps that are going to adjust and pull salt inside so that they balance. They create their own isotonic environment. Okay, quick checkpoint. In food preservation, salts and sugars are used because they create what? Do they lower the pH? Create a hypertonic environment, a hypotonic environment, or lower osmotic pressure? So what we just talked about, it's going to be B, hypertonic. Remember, the more stress we put a cell under, the higher the osmotic pressure. So stress is osmotic pressure. That's how much salt water. So it should be B, hypertonic. So they're causing the water to leave the cell. So let's talk about some other requirements for growth, things that our organisms need. So those things that we just talked about, pH and salts and temperature, those are all considered to be physical. Okay, those are all factors that affect how they grow outside of themselves. We're now going to talk about chemical factors that are factors that affect them, and they're ones that they create or have access to grow in their cells. So some chemical requirements that they need. They're going to need things like carbon. So we mentioned before that we use carbons in our carbohydrates, our lipids, our proteins, and our nucleic acids. They need those as well. They need to be able to create new lipids and carbs and all the things that they need to survive. So they're going to need a carbon source. We mentioned last time that some organisms like us are chemoheterotrophs. So we get our carbon from other organisms that we consume. Our carbon comes from our food, our salads, our meats. That's where we're going to get our carbons from. Some organisms we mentioned last time are autotrophs. Remember, autotroph means self-feeding. So these are going to be organisms that can make their own carbons, or they basically can build their own carbon molecules by pulling in CO2. So these would be things like plants, and then also photosynthetic bacteria. And photosynthetic archaea. You don't want to forget those archaea. Other things they're going to need to grow. They're going to need nitrogen. Nitrogen, remember, is found in amino acids. It's found in our DNA and RNA molecules. Most of our bacteria are going to get their nitrogens through decomposition. What that means is they're going to break down amines that are present around them. So, for example, bacteria growing in the soils, they're going to break down nitrogen that's present as either nitrogen gas or as decaying plant material in the soil. Organisms growing inside of us are going to steal nitrogens from us. That's one of the reasons why we feel tired when we're sick. We're feeding too. It's like when you're pregnant. It's like for the girls on it. It's like when you're pregnant. So you're feeding too. You're feeding these bacteria as well. Well, maybe like 2 billion, because it's a lot of them, right? But luckily they're small. Okay. Other thing that is fascinating though is these bacteria at the bottom that do something called nitrogen fixation. These little guys are really like unsung heroes in our environment. So right now we're breathing in oxygen, but in our atmosphere we also have a ton of nitrogen gas in too. Unfortunately for us, we cannot use this form of nitrogen. And as a matter of fact, most living organisms cannot use N2. Even plants can't use it. What they have to do is they have to rely, and we also have to rely on bacteria to take this gas from the atmosphere and convert it into a usable form for us, like NO3 or NO2, so a nitrate or a nitrite. That process of taking gas to a usable form is called nitrogen fixation. Farmers use this all the time. When they plant their crops, they will plant bacteria with them that are nitrogen fixers. And then these bacteria help the crops grow. So it's like the difference of after a week, you get a crop that is going to be this tall, as opposed, let's say we're growing corn, so it's like this tall after a week, as opposed to growing without our nitrogen fixers, and it would be like this tall. So we see like almost a quadrupling in size when they have nitrogen fixers nearby. Sulfur. They're going to need to be able to get sulfur for certain types of amino acids. Some of our amino acids have sulfur in them. So things like thymine has sulfur in it. Biotin, which is another growth organism, or growth protein that we need, is going to have sulfur in it. Most bacteria, again, can get these from the environment. They can absorb it as either like a sulfate or a hydrogen sulfide. They can absorb it. Sometimes they even release sulfurs. Have you ever driven by Avila Hot Springs?
Yes.
Oh, it's so stinky, isn't it? Yeah, it's like, oh, it's like you drive by there some days and you have to roll over windows because it's so bad. Those are natural sulfurs being released from underground, and the bacteria that are present are helping to convert and break those down. So on days when there's more sulfur being released, there's more bacterial activity and we get more of a stink. So you can thank bacteria and Mother Nature for that. Phosphorus. Remember, phosphates are important for ATP, DNA, RNA, they all have phosphates. So they need to have some source of a phosphorus. A lot of times they get this from bird guano or bat guano. What is that? Poop. Yeah, the bird poop or the bat poop. A lot of animals naturally, and especially we see it mostly in birds and bats, have phosphates in their fecal matter. And so again, these guys are like kind of getting rid or cleaning up stuff in our environment. So bird poop that's everywhere, no problem. The bacteria will break it down for you. They're going to convert phosphates into a usable form again. They recycle it. And then those bacteria are going to help grow our plants, which in turn we're going to consume. And it's kind of like this big cycle. If we didn't have these bacteria, we would be in trouble. Because our planet only has a certain number of phosphates, a certain number of nitrogens, a certain number of sulfate. Our bacteria keep those recycling for us. And so they kind of make the world go round, okay, without getting like too specific there. Other requirements they can have. Sometimes they need to have trace elements. So for example, they're trying to build a holoenzyme. Remember that term, holoenzyme? Where we take a enzyme when we need a cofactor. Same is true with them. So they're going to have an apoenzyme. They need to pull in a cofactor to make a holoenzyme. Some of their cofactors can be things like zinc, calcium. When we look at this, these are some of our cofactors too. So these are things that we need to consume. We're very similar to one another in our dietary needs. The big difference is that a lot of times, again, they can make their own, where we're having to consume other organisms or take supplements to get these. So these guys are going to live in the environment. They're going to help us with recycling these compounds. They're going to help us recycle things for vitamins, for amino acids, for purines and pyrimidines. Those are going to be our DNA bases that we're going to be looking at. Now, some of our bacteria and archaea that live in their environments are going to be like us, and they're going to require oxygen for survival. So these are going to be ones that we're going to refer to as obligate aerobes. That's what we are. We're obligate aerobes. If you take oxygen away from us, our Krebs cycle and our ETC shuts down. We get stuck going from 36 ATP to getting stuck in glycolysis. And how many ATP do I get in glycolysis? 2. Yeah, so I go from getting 36 for every glucose to getting 2 for every glucose. Not good. I can't live on that. Same is true with our other aerobes. They can't live on that. They have to have oxygen present so that they can do their ATP factors. How many ATP do prokaryotes get when they're done at the end of ETC? 38. Yeah, they walk away with 38. Excellent. Are those kinds of questions I can ask you on a quiz?
Yes.
Oh, yeah. I can ask you what's going in. I can ask you what's coming out. I'm not going to ask you all the in-between steps, but I do want to know when I walk away how many ATPs I have, how many NADHs I have, how many FADHs.
Yours is 36 because we use 2 to move it from our computer, and that's what we did.
Beautiful. Did you guys all hear that? Eukaryotes get 2 less ATP. We get 36 because we have to pay a penalty to move to our mitochondria. So that's the reason why we get 36 and they get 38. Excellent. All right, so again, those guys are going to be considered obligate aerobes. We're considered obligate aerobes. There are some other organisms, though. There's a few out there. For example, staph, E. coli, ones that we're used to, are all of our normal floras, that are going to be referred to as facultative anaerobes. The term facultative means that they can turn it on or off. Okay, so they can be there or not. Remember, we had facultative halophiles. I told you those were pumps the staph could turn on to adjust their salt, but they're not stuck on all the time. Okay, facultative anaerobes are ones that can switch from living with oxygen to living without it if oxygen isn't present. So again, they can turn on different enzymes and do different pathways. A lot of them will just stay in fermentation. They'll only get 2 ATP, but they're a single cell. Some of them don't need more than that. They just grow a little slower. Some will switch to a completely anaerobic pathway and use a different electron acceptor for their bowling ball, for the one that's pulling everything towards them at the end. And in some organisms, the ones at the bottom, these are considered to be obligate anaerobes. These are ones that find oxygen as very, very toxic. Oxygen, as we've mentioned, likes to steal electrons. We've talked about that, right? It's the biggest, baddest electron pooler. It turns out that we have enzymes that kind of neutralizes oxygen when it starts to get a little too crazy and it tries to steal them too fast. Our obligate anaerobes lack these enzymes. So when oxygen gets a little crazy and starts stealing electrons, these guys don't know what to do. They can't calm it down. And oxygen keeps stealing their electrons and basically causes them to fall apart. Kind of similar to what happens to us when we get an acid burn. Remember I told you an acid burn is from electrons being stolen? Same idea. It basically causes a burn from inside out because the oxygen's not going to be able to be stable with them. So if we were to put organisms into categories, just like with temperature, we can come up with 5 different categories. It's kind of nice to know the magic number is always 5 for this chapter. Okay, we have oxygen required on one side. That means I need it for life. Okay, obligate aerobes, that would be us. And another group of organisms referred to as microaerophiles. Okay, these guys are funny. They have to have oxygen present to live. However, they need less oxygen than what we usually get. So let's say, for example, with every breath that we breathe in, we get 100% oxygen. Okay? Microaerophiles, they can't live on 100% oxygen. They can live on like 70%. But if I took the oxygen away, they would fall apart. Right? They have to have oxygen present. So these are going to be organisms that a lot of times we find deeper in soils. So they're getting some oxygen coming through. These are organisms that we find a lot of times in deep lungs. So like way, way deep in our lungs where we don't take as big of a breath. So they can grow in the microaerophilic conditions. Now, these guys, again, are a little odd. And the reason that they can't survive high amounts of oxygen is because they produce enzymes that help slow that oxygen down when it gets a little crazy. But we produce more enzymes, they produce less, so they can't keep up with oxygen when it's at a high level. Does that make sense? It's kind of like, if you're trying to run a daycare and you have snacks for kids. If you have tons of snacks, the kids are happy, right? That's like our obligate aerobes. We have lots of snacks. Okay, now I'm running a daycare, but I'm going to run a lower number of snacks. And if I give the kids too many snacks, they're going to throw up. So I want to keep them at a 70%. Does that make sense? That's kind of how these guys are. They get sick if it's too much. All right. Again, I give you lots of analogies, and because I'm a mom, I give you lots of kid analogies. Sorry, I apologize for that. Okay. On the other side, we also have these groups of organisms that don't require oxygen. Okay. So obligate anaerobes, they cannot have oxygen to survive. Like if they get oxygen, they die. We mentioned an organism in here that we have to watch out for on rusty nails. because it can hurt us if it gets inside an anaerobic area. Yeah, do you guys remember what it was? You can say it louder, tetanus. Yeah, Clostridium tetani, right? Clostridium, that entire genre, Clostridium, there are always going to be obligate anaerobes. They do not like oxygen. Any of them. Clostridium difficile, Clostridium, which one do I want to talk about now? Tetani, they do not like to have oxygen. Facultative anaerobes, remember these are the guys that can switch. They can go with or without, they don't care. They'll figure it out. Error-tolerant anaerobes. These are organisms that do not need oxygen to survive, okay, but they can tolerate it. So what that means is they have the enzymes to neutralize oxygen. It's not going to cause them to die if it's around. But they also do not use it in any of their processes. So if I take it away, they're fine. If I put it on them, they're fine. They could care less. So error tolerant, they're going to react, whichever. So let's look at why this happens. What is it about oxygen that makes it so, volatile? Well, it turns out that oxygen, as we know, in our environment usually exists as O2. Have you guys heard that? O2? Yeah. And the reason it exists as O2 is because, remember, it's trying to fill electron shells. And oxygen, a lot of times when it's in the atmosphere, will bond with another oxygen molecule. It's like, oh, hey, are you unhappy? I'm unhappy too. Let's get together and share. Okay. But is oxygen a good sharer? They're not really great sharers, right? And so now they found that they're equally pulling the electrons, and so neither of them are super happy. So what do? They go, well, we're both miserable. Let's go ruin somebody else's day. Have you ever run into somebody who's like that? Somebody who's miserable and they have to ruin your day. That's kind of what happens. So these O2s are both miserable because they're pulling the electron back and forth. They're fighting over it. So they're like, you know what, this is hard. Let's just go steal an electron from somebody else. And so what they do is they start to look around and see who they can steal from. Usually they find hydrogen nearby and they're like, hydrogen's always a sucker. Let's go ahead and hit hydrogen up. And so they go and steal its electrons. That's how we end up with something called H2O2. We refer to that as hydrogen peroxide. So we actually will get a natural hydrogen peroxide that'll form. Now, when this is inside of our cells, this is not good. We don't want to have hydrogen peroxide inside of our cells. We talked about hydrogen peroxide when we put it on our skin, right? It bubbles. And I told you the reason it bubbles is we have enzymes that neutralize it, that break it down. So it turns out we produce an enzyme called catalase. You can see that there, it says catalase. We produce an enzyme called catalase that neutralizes those toxic forms, and they turn things into harmless water, and they put the O2 back into O2. They calm it down, okay? We also can have now this unstable form be broken down by another type of enzyme. This is called glutathione peroxidase, and it can take apart the enzyme and put it back to just water. It can make the hydrogens and the oxygen separate to become water. And I can release regular oxygens, or I can hold on to them and use them somewhere else. Now here's the deal of why this affects our organisms. If we're an aerobe, we make all of these enzymes. Superoxide dismutase, catalase, peroxidase. We make all of those. So we can neutralize oxygen, we're good. If we are a facultative anaerobe, we make all of those. If we're a microaerophile, we make one of them. This is why they can't handle too much oxygen. They have to go kind of slow. So they'll either have the dismutase, and then a lot of them will have maybe a catalase, but they won't have a peroxidase. So they can handle some, but not too much. Okay, if I give them too much, they break apart. Obligate anaerobes, they have none of these. So obligate anaerobes, they are just, at the mercy of the oxygen. They fall apart. And then if I'm going to be a error-tolerant anaerobe, I produce all of them. I can neutralize it. All right, that makes sense what's going on behind the scenes. All right, so how can we tell if an organism needs oxygen to grow or hates it to grow? Well, it turns out we can use a special type of media. It's referred to as fluid thioglycolate. We refer to it as FTM in the business, fluid thioglycolate medium, FTM. Okay, FTM is a broth, but it's kind of a thick, soupy broth. When they make this broth, they put in a little agar, just a little, to make it a little thicker. Okay, so it's not solid, it's just a little. For example, a Petri dish has about 12 grams of agar to make it solid. Okay, this tube has about 2. So quite a lot less, but just enough to make it a little soupy. So it's almost like a pea soup consistency. Now why that matters, at the top of the tube, I will be able to have oxygen present. So I can do oxygen exchange. So if I'm an aerobe, an obligate aerobe, I'm going to be living at the top of that tube. Okay? Then, as I move farther down in the tube, the base of the tube has no oxygen. It's so thick, we get an oxygen gradient. And so as I move down, I get less and less oxygen. So if I was to draw this tube then, I would say that I have high O2 at the surface. And as I get down here at the bottom, here, I'll push that back so you guys can see, I have no O2. So at the bottom of the tube, no O2, high O2. Okay? We call that an oxygen gradient, meaning that we have different levels of oxygen in the entire tube. Okay, so when I grow my organisms, depending on where they grow, I'm going to be able to tell what type of organisms they are. So let's look at letter A. Letter A says it's an aerotolerant anaerobe. Can they handle oxygen? Yes. Remember, aerotolerant means they can handle it. Do they need oxygen? No. Remember, they don't use it, but it doesn't hurt them. So what we see is they grow throughout the tube, but do you notice how there's a little gap at the top? They don't grow like really fast when there's oxygen around, so that tends to slow their growth down. It doesn't really like hurt them though, it just kind of slows them a little. So they tend to hunker more in the anaerobic areas because it's easier for them. They're not having to create enzymes, but they can grow all the way up towards the top of the tube. They're tolerant. B, check out my obligate anaerobes. Are they hunkered at the bottom or what? I mean, we can tell where the oxygen stops, right? They're all like at the bottom, trying to hide from it. They can't grow any higher than that because there's too much oxygen. So we can tell that they're obligate anaerobes based on where they're growing in the tube. Let's look at C. C is a facultative anaerobe. They grow faster with oxygen. Do you see how at the top I have a heavier growth? It's almost like a mushroom cloud. The reason for that is at the top they're getting 38 ATP. At the bottom they're getting 2 ATP. So they grow a little slower. Still grow, but just not as fast. D, microaerophiles. Check them out. They're growing in the oxygen range. You can tell that because we can look at B and tell where the oxygen runs out, right? So I can look at D and say, yep, they're above where the anaerobes are growing, but they're not at the surface because that's too much oxygen. So they're growing right in that middle ground, like around 70%, maybe even closer to 60. And then check us out. This is where we would be growing. Obligate aerobes, look at us. We're all hunkered at the top, gasping for air. It's like being in a pool. You're all floating at the top, like, and if you get pushed under the water, you're going to not make it, right? So look at them. They're all fighting for that upper space where oxygen is plentiful. All right? So just setting up an FTM tube, fluid thioglycolate tube, that will tell us whether or not we have an aerobe or an anaerobe, or what kind of aerobe or anaerobe we have. That's a pretty handy little tool. Okay, some microorganisms use gaseous nitrogen directly from the atmosphere in a process called what? What did we say that was called when they take nitrogen and convert it?
Nitrogen.
Excellent, yes, excellent. C, nitrogen fixation. Excellent, they fix it for us. Isn't that nice? This, again, is kind of an unsung thing that bacteria do, because if we didn't have them doing this process, we would be in trouble. We need to have these guys around doing this for us. This organism is killed by atmospheric oxygen. If I expose it to oxygen, it dies. Which organism is it? C. Obligate anaerobe. So if I expose it to oxygen, it's going to die. Obligate anaerobe. How would one determine whether a microbe is a strict anaerobe or not? I have this microbe I've grown. How am I going to decide what kind of oxygen it likes? Excellent. I'm going to grow it in FTM media, and I'm going to look at where it grows. Does it grow at the top of the tube, the middle of the tube, the bottom of the tube? That's going to give me information. Does it make a mushroom cloud? How is this organism growing? That's going to tell me what kind of anaerobe or aerobe it is. All right, let's talk a little bit about how we grow these organisms in our media. So we've been using media since the beginning of lab. We've been using plates and tubes and things. Let's talk a little bit about what's inside of that media that we use. It turns out that culture media, what we use to grow our bacteria, are basically just nutrients that we give them to be able to support their growth. Now, our media has to be sterilized because we don't want to have anything growing in there before we start. Can you imagine if we gave you Petri dishes that weren't sterilized? They'd be covered in mold and gross stuff, right? If you ever want to see how gross our lab is, take a Petri dish and just leave it without a lid for like a day or two. We will have tons of growth. We did that one semester. We can do it again if we get bored and never try it. Just put a Petri dish in the corner and everything in the air falls on it. Ooh, we should actually try with our air purifier, see if it's making a difference. Ooh, that might be fun. We can put one close to the purifier and one close to the door. What do you guys think?
Yeah, let's do it.
Okay, we're going to do it. I'm going to make a note to myself that we're going to do that with some NA. So nutrient agar air test. Okay, we're going to have some fun. We're going to do that today. So today. Okay, so we're going to see what's growing in our room. Oh, it's going to be so disgusting. Oh, but it's okay. How come we're able to handle all this stuff floating in the air? It's not all bad. It's not all bad. Excellent. Most of it's just harmless. And our body's immune defense system, it is spot on. It's able to help us filter a lot of this out. Okay, so we want it to be sterile. And the way that we sterilize it here is we use autoclaves. We'll talk about the autoclave process. We basically superheat the media. And that kills everything in it, including endospores. And that's the big one. We're going to add an inoculum, where we add microbes to something. And then we're going to culture, which means we're going to grow the organisms. So we've been using these terms in here for a while. So some of this chapter is going to feel familiar, which is nice. Same thing with chapter 3. Remember the microscopes felt familiar? We're running into the same thing here. So one of the things we're going to use is agar. And we're only going to use agar when we want solid media. The one exception to that is FTM, that fluid thio broth that I just showed you. But again, it has just a teeny tiny amount of agar to make it like pea soup, okay? Otherwise, if it's a broth, no agar, okay? This agar can be used to solidify our media so that we can get cool stuff like slants or deeps or Petri dishes. And the beautiful thing about it is at this point in time, there's not a single organism that can eat agar. I'm sure one day we'll find one, but as of right now, none. They don't have the enzymes to break agar down. Thank heavens, that's why it works so well. As I mentioned before, we used to try gelatin, but the problem we ran into is a lot of bacteria have an enzyme called gelatinase, which means that they eat gelatin. They're like, oh, thank you, this is nice, and they turned it into liquid again. All right, that wasn't helpful. So agar, luckily, there's no agarase. Thank heavens. Whew. Okay. The other thing about them is that with agar, I can liquefy it when I boil it. So when I make my agar, when I'm making Petri dishes, I'll throw in my starting media. We've purchased the media ahead of time from a company. It's kind of like a cake batter mix. You ever bought like a box of cake batter or a brownie mix? That's basically what it's like. They've already put everything in it. We dump it into a big liter batch. We boil it up to mix it. When we boil it, any agar in there is going to be liquefied. We autoclave it, we pull it out of the autoclave, let it cool for a little bit, and then we pour it in Petri dishes or pour it to make slants before it gets to 40 degrees. And the reason for that is once we get to 40, it starts to solidify again. And if you're not pouring Petri dishes fast enough, you'll turn your flask over and it's just like solid in there. Or you get a big chunk that falls out. That's the worst, because that's just like depressing. So we have to pour it before it hits 40. So again, here's a picture of what we used to use, the gelatin, and here's a picture showing gelatinase in action. So in B, we have gelatin present that's solidifying the media, and our organism that's growing on that one is not producing gelatinase, so it's staying solid. But check out organism A. Organism A made gelatinase, and that means that even though it started in a solid, its media originally looked like the tube in B, it produced that enzyme and it liquefied it. So when you come in the next morning, it looks like a big pile of liquid. All right, again, not super helpful. So that's why we rely on agar so much. We actually do use gelatin nowadays for a test for gelatinase. We look to see if they make that as an enzyme. It tells us a lot of times if certain organisms are pathogens. It turns out that a lot of pathogens make gelatinase. When they're growing inside of us, they are using us as a gelatin source. Isn't that vicious? They're so evil. They take all of our nutrients. And so one of the things that we can create for them is a gelatin type material, and they will use that as a food source. Stinkers. The other thing about media is we can use different types of media for different types of tests. For example, I told you we can buy our media ahead of time. It's kind of like a cake mix media. And the first type of media we can choose is one that's going to be very expensive. It's called chemically defined media. This media is pricey because when it's made, there's a lot of time and effort that goes into it. Everything that's in this media is carefully weighed. We know exact amounts, and we know exact purities of everything used. So for example, our cheaper media will use something like animal extract. Okay, that sounds weird. But an example would be us buying a steak. Okay, in that steak, there's animal extract. What is that? It's animal proteins and lipids and DNA that's in the steak. Do I know exactly how many proteins are in there? No. Do I know exactly how much DNA is in there? No. I don't know. It's just whatever the animal had, right? Some will have more fat. We see that. Some have more lipid than others, right? Okay, I don't have exact counts. And when it's complex media and I don't have these exact counts, I don't care. However, chemically defined media would never use like a steak as its basis. Instead, it's going to measure ingredients out perfectly. So I'm going to know exactly how many proteins are in there, exactly how many lipids. Now you might be asking yourself, why? Why do I have to know exactly what's in there? Turns out we use chemically defined media when we're testing an outside source for something. An outside source would be something like an antibiotic. So if I want to see if an antibiotic can kill a bacteria, I can grow it on a chemically defined petri dish with that antibiotic. And I know the only variation from this dish to the next is the antibiotics reaction. Does that make sense? If I ended up with petri dishes that had more protein in one and less than other, I could get variations that had nothing to do with my antibiotic. So I have to control everything so that I can test antibiotics. Okay, so it's a very defined test. Complex media, that's what we usually use in here, nutrient agar, TSA, all of these are complex media. And they just have extracts in it. There's like a piece of steak blended up. There's like some spinach blended up. Some seaweed blended up. So it's a little bit of everything. I don't know the exact proteins. I don't know the exact lipids. I don't care. Just like when we eat, we don't care. It's not like when I buy spinach at Costco, I'm sitting there going exactly how many and what type of proteins are in here. There's a little bit of general information on my nutrition label, but it's not exact, right? There's always variation from one lot to the next. So, an example of a chemically defined medium. So again, this is used for antibiotics. I know exactly how much sugar is in there. I know every single nutrient in there, ammonium phosphate, and I can draw it out, for example. Sodium chloride, magnesium sulfate, potassium phosphate. These are all pure forms that I have, and I know exactly what's in there. And again, the only variation I'll have then is the antibiotic that I'm testing. Here's a picture of antibiotic test plates in action. So what you're going to do is you're going to take a petri dish, You streak your bacterium all over it, make what's called a lawn. We call that a lawn of bacteria. It grows evenly everywhere. And then we add these discs that have different antibiotics on them, and we look to see which one is allowing growth and which one is inhibiting growth. You can tell the ones inhibiting growth because they have halos around them. So based on what you see here, if you're a patient, should I give you antibiotic A? No. Do you see how the bacteria is growing right up to it? Yeah, that would be a poor choice. Have you ever started an antibiotic and had the doctor call you a day or two later and switch your antibiotics? Yeah, that means you have a good doctor. What that means is they took a sample, maybe it was a throat swab, they took a sample and they cultured it and did this test. And they gave you the most typical antibiotic, but they confirmed that with one of these tests, and they found that your microbe was resistant. And so then they're going to switch you and put you on an antibiotic that would work better. Which one do you think would work better on here? E. E. Does everybody like E? Yeah, if I was a patient, I'd want E. I'd be like, that's the one I want. Give me that one. So they'll call and switch you to that one. Okay? All right. This is a picture of complex media. So in here, I have some beef extract. I mean, some blended up steak or blended up hamburger. I don't know exactly how much is there. I don't really care. I mean, I know how much I've added. I've added 3 grams, but I don't know how much, protein, DNA. I don't know what's there. I don't know what kind of proteins are there. There's just proteins, and the bacteria are fine with that. Sodium chloride, peptone. Peptone is a milk protein. So it's found in dairy that we dry it out. We dry out the milk, and we get these proteins that are left behind. Agar, water. So I'm measuring it, but I don't know exactly what the counts are inside of my measurements. Okay? And again, it's not the bacteria don't care, just like we don't keep track of every single thing in our food. So this media is the one we typically use. The complex medias are typical. And the reason for that is it's going to be cheaper because they're not having to have somebody measure everything out meticulously and run analyses on it to make sure we have proper amounts. It's easier to produce. And again, it comes to you kind of like a cake mix. So you just add water and you're good to go. This can also be adjusted to grow target organisms. If I'm trying to grow staph, I'm going to add a whole bunch of salt. If I'm trying to grow gram-negative, I'm going to add some crystal violet. Do you see how this works? So we can fix our media by throwing stuff in there to make it selective, differential, or both. So that's what we do with our complex media. We would not be doing that with anything that would be with our previous media, our chemically defined. We wouldn't be doing that. We don't change it. Okay, there's another type of media. This is the FTM, fluid thio, and we call this in the biz a reducing media. What this means is it's media that has less oxygen in it. So remember FTM, our fluid thios, the bottom had no oxygen, right? And so this is media that I can immediately put an anaerobe in, and they could grow right away. As soon as they get to that bottom part of the broth, they can start to grow. So we refer to that as reduced, meaning that we pull the oxygen out of the bottom of it, all right? The way we do that is when we heat it, the oxygen kind of puffs out when it's being heated through the boiling process. And then we allow a little oxygen to get back in for the head space, the top part, and then we tight cap it. And then that way there's only a little oxygen in there. Not a whole lot, so my anaerobes can grow. We say that it's been pre-reduced, that means it's been boiled to push oxygen out. That's all that means. We also sometimes, though, need to grow anaerobes on a petri dish. Remember we said petri dishes are better to be able to see individual colonies? So we run into instances where I need to grow an anaerobe on a petri dish. This is the way I can do it. We're going to do a lab in here that's going to do this. We're going to streak our plates with our anaerobes, and we're going to put them inside of a jar with a pouch that's going to pull the oxygen to it and destroy the oxygen. So this is the way that we can grow our strict anaerobes. Luckily for us, most of our anaerobes will make endospores. So when we're transferring, they're dying. When we're streaking our plate, they're dying. When we put them in the jar, we're waiting for the oxygen to get absorbed, they're dying. But as soon as the oxygen is gone, guess what their endospores do? They come back, they go, woohoo, and they hatch out, and they start growing again, so we get them back, okay? So that's why we're able to use Petri dishes, thank heavens, so that we can see the individual colonies. We have some organisms that will grow in anaerobe chambers. Anaerobe chambers are basically glass tanks with sleeves inside of them. You put your hands in the sleeves, and you have tanks attached to them that are pumping in things like CO2s and nitrogens, anything that's not oxygen-based, okay? And so that means that in that tank, it's completely anaerobic. If I'm working with an organism like Clostridium that I want to study for a test and I don't want to have any death at all, I can use an anaerobe chamber, which means they never get exposed to oxygen. They never even make their endospores. They just stay like this. Isn't that nice? So I can put them in a jar in here and everything. So they never get exposed. I will tell you there's a couple of drawbacks to this. If you are latex allergic, I'm one of those lucky latex people, those sleeves usually have a latex band, which means you always have to make sure that you're wearing long sleeves if you're going to work in these, because otherwise you come out with cool tribal tattoo patterns that are from your skin reactions. I'm telling you from experience. The other thing is always use the restroom before you get one of these things, because that's what's going to happen every time. The coffee that you just drank will kick in right when you're starting to streak. You're like, oh my gosh, because you can't get out. You got these glove things on. You got to finish what you're doing. All right. So those are life lessons that I will share with you. All right. Capnophiles. These are a little group of organisms that are special. And the reason that they're special is they like to have CO2-rich environments. If we were going to put them in with the other 5 categories we talked about, they would go with the microaerophiles. Remember we said less oxygen? Yeah. Capnophiles are ones that love CO2. So these are going to be organisms that we're going to grow, for example, in what's called a candle jar. This is really cool. You take a big mason jar, you put your Petri dishes in there, you light a candle, and you screw the lid on top. What does a candle or a flame need to burn? Oxygen. So it's going to burn and use all the oxygen, and then it'll extinguish itself when the oxygen's gone. So the oxygen will be there for a while, allowing the organism to grow, and then it'll go out, and then that'll stop its growth. Yeah, cool technique. That's awesome, right? Okay, where do you think I'd find organisms that like a lot of CO2 in our bodies? In our lungs. Yeah, in our lungs. Sometimes high up in our nasal passages. They like that CO2 that we're breathing out. So these are organisms, again, that they like to have a little oxygen, but not too much. So these guys like to balance it. It's like 70% oxygen, 30% CO2. They like to have that balance. If you ever go to a hospital and go into their micro lab, they will always have a CO2 incubator. Always. And it's an incubator that will look just like the little ones we use, but it'll be hooked up to a CO2 tank. So it's pumping in pure CO2 for those guys. And they love it. They'll grow very quick. That's where you grow like your TB strains. They love to have that CO2. It helps them grow a little faster. Selective media. We talked about this in lab. Selective media, remember, is where we're trying to grow what we call a target organism, the organism that we want to grow, and we're trying to kill off or suppress organisms that we don't want. This gets rid of a lot of background noise. In other words, I get rid of all the background normal flora. I get rid of all the background bacteria and hopefully just grow my pathogens. That's the goal. It makes it easier. Okay, so we had some selective media in our lab. We used mannitol salt agar. Here's a picture of mannitol salt agar. Remember our plates were red when we set them up? There's Staph epi growing. It's still kind of a reddish pink. Look at Staph aureus. What color did it turn the media? Yellow. We should see that today. Okay. So other types of selective media. Sabdex, that's used to grow yeast. I mentioned way back that yeast like to grow in acidic conditions. So Sabdex is one that is acidic. So it'll encourage yeast to grow and discourage bacteria. So Sabdex is going to be acidic. Brilliant Green and McConkey, they're ones that use dyes to kill off background flora and only allow the pathogens to grow. Differential media. Differential media is one that shows us a difference between multiple organisms. Now, sometimes our media is differential and selective. Sometimes it's just differential. This is a picture of a media that is just differential. It's not selective at all. This is a blood auger plate. Doesn't that sound horrific? Just in time for Halloween, blood auger, okay? It's like, Dracula's plate. So what's going on in here? Well, it turns out that media manufacturing companies, they actually, almost all of them, have farms in Montana. And guess what they grow on these farms? They grow sheep, they grow horses, they grow them, it's like crops. Here's a crop of sheep, crop of horses. No, they farm them, okay? Sheep, horses, pigs, anything you can imagine. But here's the deal. These are probably the most pampered animals you're ever going to meet because they use them for blood donations. We learned a long time ago, back in Koch's age, that if you use slaughterhouse blood, the animals are stressed and scared. It changes their proteins. Like when we get stressed and scared, it changes our bodies. And it actually makes it so that when you try to grow bacteria on those blood plates, the bacteria won't grow. It's almost like they pick up on the stress. There's stress hormones and enzymes in there, and it stresses the bacteria out. So you want to have happy cows. You want to have happy sheep. You want to have happy horses. So what they do is they all have colored tags, and they rotate them. So they come in and they'll do a bleed, just like a human doing blood donations. They'll do a blood donation, and then they'll let them go for a month, and then bring them back in and do another blood donation. So it's not like this horrific thing. So here's some blood auger plates. Again, it has red blood cells in it. And we see here different types of enzymes producing that are affected in the blood. We have alpha hemolysis. Hemolysis means breaking the blood cells. We have alpha, which is shown here, the alpha with the little A sign. We have beta, and we have gamma. Gamma is the little B at the bottom. Notice they all look different. So let's talk about what's going on here. Think of the red blood cells as kind of like a hard-boiled egg. Just picture that in your mind, okay? Alphas are taking that hard-boiled egg and hitting it on the counter and cracking it, okay? They're not able to remove the shell, but they're able to just crack it and weaken it. So then they can pull some nutrients out of the blood cell. So what we see with an alpha is that the blood cells show a little damage. They're a little green colored, but they're still intact. Just like taking a hard-boiled egg and shattering it on the counter. So the shell's broken, but it's still intact. The betas, they're taking that hard-boiled egg, they're hitting the shell, and they're pulling the shell completely off and eating the egg. All right? Beta hemolysis, as you can imagine, when you get it, it hurts. They're lysing, they're popping open your blood cells. It hurts. Anybody in here ever had strep throat? Yeah, strep throat is beta hemolytic. When I take a throat swab of a patient that would come in with strep throat, we'd throat swab them really quick. The enzyme that we're looking for on a rapid test is the one that does the beta hemolysis. That's your rapid test. That's your 5-minute. If you have kids, they should never only do a rapid test. They should always culture as well. And the reason they need to culture is kids have smaller throats, so they're going to have less strep, which means I might not get enough for a rapid reaction, so I have to grow them and then look for hemolysis. That beta hemolysis, when I hold this plate up, I can see through it. It's like a window. This is why strep throat hurts so much. It's because your throat is raw, because it's popping open your blood cells. Ouch, not a nice bacteria to have. Gamma means that they're not hurting our blood cells at all. So we have a lot of bacteria that are in the gamma range. They don't really even recognize it as a food source. It's just like if you have somebody that doesn't like hard-boiled eggs. They walk right by them. They don't even want them. All right? We also can have enrichment culture. Enrichment culture is where we are encouraging an organism to grow faster. For example, we're giving it its very favorite food. Like for me, if I was in an enrichment culture, I would have Cheetos and Starbucks coffee. Okay, you'd be giving me my favorites. All right, Cheetos and Starbucks. All right, these are going to be ones, for example, if I'm trying to grow an organism maybe in a fecal sample, which is going to have a lot of background, or a soil sample, a lot of background, and I want to grow my pathogens or my organisms of interest, I give them whatever I know they like to eat so that they grow a little faster. Now, the key between enrichment and selective. When I'm growing with selective, I am killing off some organisms. So for example, my staph, I killed them off with salt, the other organisms. With enrichment, I don't want to kill them. I don't want to get rid of those background, but I want to have one grow faster. Okay? It's like if you were a bird, you'd be feeding one of your babies more than the other. So that baby bird that's getting more food would get bigger, but you're still not letting the other one starve. It's just that you have a favorite, right? I always swore that my mom had my brother as his favorite. That's why he's 6'4" and I'm tiny, because she fed him. It's not true, but I'd always tease her with that. Okay, so the idea though is you're getting different sizes or you're growing faster. Why would we not want to kill off everything? Can you think of a reason? Maybe we have a patient come in that's having diarrhea, and we think it's salmonella, so we're going to put it in a media that's in enrichment. Why would I use this over a selective? Can you think of any reason? So remember, with enrichment, I'll still see everybody that's there eventually. But if it's salmonella, it'll grow faster. I'll see it first. The reason I would want to use an enrichment is because maybe my patient doesn't have salmonella. Maybe they have E. coli 0157. So I would still be able to grow it. It would just grow slower. So I would still get potential pathogens. I'm not getting rid of them completely. I still can grow them, but they'll grow a little slower. So I would hold my plate a little longer. Okay. We also run into problems, though, with some bacteria that we just can't culture in a petri dish or in a broth. These are ones that are so picky. They have to be inside of an animal host, okay, almost like a virus. So, Mycobacterium leprae. What do you think that causes?
Leprosy.
Leprosy. Yeah, leprosy. Do we still have leprosy nowadays? We do. We still do. Remember, smallpox is the only thing we ever got rid of. So we still have leprosy. We can treat it faster. So leprosy is an organism that attacks the cartilage. And that's why if you see pictures of people who used to have leprosy, a lot of times they're missing noses, they're missing parts of their face, their ears. Wherever we have high cartilage, it would dissolve those tissues. So we do still have it today. We just can treat it. So we can save their noses, save their ears, but they'll still have the infection. Treponema pallidum, we talked about it before. Where does it grow? Syphilis, yeah, it's an STD. So it's going to start in the genitalia and then it can work its way to the brain maybe. Okay, these bacteria, I can't grow them artificially. The way I can grow them is with some sort of a host. And this is where it gets funny. Trying to figure out what host to grow them in is always a hard one. Mycobacterium leprae, the one host other than humans that we can grow it in, are you guys ready? It's an armadillo. How did they figure that out? I always wondered, did they start with the A's and just work their way down? Aardvark, no. Antelope, no. It didn't work. We get to armadillo, ding, we got a winner. Yeah, they can grow it in armadillos. How strange is that? How random is that? Okay, so Mycobacterium leprae is grown in armadillos. Treponema pallidum is grown in bunnies, in particular male bunnies in the testes. So we have to, have the right host to be able to grow the right organisms. Okay, to cultivate anaerobic bacteria, one must use a special media called what? What do we call FTM? Reducing. Excellent. Hey, reducing. So reducing media. Remember, it has been reduced. We don't have as much oxygen in there. The culture medium that is designed to increase numbers of desired microbes to a detectable level. Now notice I'm increasing. C, enrichment. Good, enrichment. Now, what if I said the culture medium designed to only grow desired microbes? Excellent, that would be selective. B, so only would be selective. If I'm trying to grow target organisms faster, detectable, then that would be my enrichment. Let's talk about some biosafety levels. We mentioned BSLs with our very first lab, and I told you the organisms that we're working with in here are fairly harmless. And I said, we're going to be working with BSLs of 1s and 2s. And you went, okay, well, we're going to talk about what that actually means today, all right? So biosafety levels are assigned for every organism out there. And it tells us how pathogenic they are. how virulent they are. We've used those terms before. What do I mean by pathogenic? Allan? It causes disease. Yeah, so pathogen virulence. It's how easy it is to cause a disease. So in other words, is COVID-19 virulent? Yes. Yeah, it causes disease pretty easily, right? So it is going to be virulent. It causes a disease easily. All right? Okay. So we are going to have different levels assigned depending on risk. In our lab, we tend to use mostly level 1s, BSL 1s. BSL stands for biosafety level. So we have BSL 1s where the organisms are not easily spread or acquired. So for example, E. coli, Staph epi, Bacillus megaterium, Bacillus subtilis. These are organisms that we can handle, and as long as we're not drinking them, which I don't know why we ever would, we're going to be fine. If we drink them, we're going to get an upset stomach. Okay, but we're not going to die from it still, even from that. We just have to use precautions in the respect that, you don't want to walk out of the lab without washing your hands, things like that. Okay. Number 2, BSL tubes. This is where we're going to go up a notch. You're going to need a lab coat. I have this for lab coats in here because I want to protect your clothes, and plus I think it makes us look cool. So that's why we do it, all right? So in this lab, you'd actually need a real lab coat, okay? And that lab coat would be very nice. It wouldn't be like our ones that we throw away. So a nice lab coat, always gloves, whenever you're handling them. And you'd also want to wear eye protection, some sort of a glass goggles or some sort of something over your face. These organisms can cause mild disease, but they are difficult to aerosolize. Now here's what that means. You know when we take our tubes and we flip our bacteria over, or when we flame our loops? If I do that quick flip and then take the lid off, if I had an aerosolized bacteria, it would pop up in the air. We don't have those. Our bacteria are heavy and dense, so that's why they go to the bottom of the tube as it is. When I flame my loop, if I had an aerosolized bacteria, as soon as it got hot, it would go and it would release a cloud that could make me sick. Our bacteria don't do that. Okay? So the aerosolized ones would be things like MRSA. So that's not easily aerosolized, it's heavy. Things like Lyme disease, check it out, influenza, and HIV. Do those still cause illness? Yes, they do. But it's harder to contract these when you're working in a lab, okay? So in other words, can I get MRSA by sharing safety equipment? Like we see that a lot of times in sports, equipment that's rubbing on your knees or whatever, pads. Yeah, you can definitely get MRSA that way because it's right against you and it's being rubbed into your skin. In a lab setting, are you taking your test tube and rubbing it on your skin? No. So it's not easy to contract it. All right, it's also not light and aerosolized, so I don't have to worry about breathing it in, okay? HIV, do I have to worry about HIV when I'm out in the wild? Yeah, you have to have protected sex, right? You have to make sure we're careful. But in a lab, it's not like you're going to be doing anything weird with test tubes, okay? So we don't have to worry about it, okay? I mean, you shouldn't be. So we don't have to worry about you getting HIV when you're working in a lab, okay? Influenza. Influenza is one that most strains can be aerosolized when they're coughed out or when they're sneezed out. In a lab, does your test tube sneeze or cough? It just sits there. So that's why it's considered to be a level 2. COVID, we were not sure what to do with it. We still don't know. It goes between a 2 and a 3. It's almost like we need to create a separate category, like a 2.5 for it. It's not the easiest aerosolized, but it also sometimes can be passed through, like if you touch it and then touch your eye, which you shouldn't do, or touch your mouth, it could be potentially passed that way if it's a high load. But you have to have the contact source right there. If you have it on a paper and you hand it to me, 5 minutes later it's dead. So like I said, it's right in between. It's a weird thing, so we're still not sure how to handle this one. Let's talk about BSLs of 3. This is where we start to up the stakes a little. These are ones that we have to work with carefully because they are light and they can aerosolize, meaning I could potentially breathe them in. So I want to work under a safety cabinet. A safety cabinet means I work in something that kind of almost looks like that hood there, and the air would be pulling away from me. So this hood is like between the 2 incubators. The air circulates and pulls away. So if anything aerosolizes, it goes away through a filtration. all right? It never gets close to me. I also am still doing all of my level 2 requirements, the lab coat, the goggles, the gloves, all of those stay. They just keep adding, all right? I also am going to have special coveralls or scrub suits that are going to stay in that room at all times and then be incinerated when we're done. And usually these rooms have key card access. We're being much more careful with who can come in and who can leave. Okay, a lot of these organisms can be fatal. So some of our organisms in here, for example, anthrax, it can be fatal at a high load. So what we are able to do is we're able to grow them because we're careful to not reach the fatality load or the fatal load. We also have vaccinations in place for all of the organisms that are class 3s. So for example, I can work with Salmonella typhi, which is a nasty organism that can aerosolize because I can get my typhi shots. Okay, so there's things that we can do, typhoid shots, to avoid getting these organisms. West Nile virus, there are treatments for it. Not necessarily a vaccine yet, we're almost on the edge of that, we're still developing it, but there are treatments for it. Biosafety level 4s, this is where you go if you like excitement, okay? You would never find me in a BSL-4. BSL-4s are ones that have negative pressure. Okay, what that means is that if the door opens, the air pulls into the room because we never ever want the air from this room to leave. So it's negatively pressured, it'll pull air into it. The air can't leave it. It's got air that's filtered twice. These are organisms that aerosolize like that, and they are all fatal. Okay, you don't want to get these. These are ones that you want to definitely be careful with. These are things like Ebola. These are things like Marburg, which is a type of Ebola, Lassa fever. These are our hemorrhagics. These are the ones that if you breathe them in and get exposed, you can potentially die from bleeding out. You'll be bleeding from every orifice. It basically liquefies your tissues. Not a pleasant feeling, okay? If you get a rip in your suit, if something weird happens, and here's a picture of BSL-4. You can see they're all wearing what are called bunny suits, these big suits. This is like what you see in the movies, right? They're hooked up to these hoses that's pumping oxygen into them. They're staying in these rooms. There's a quarantine room. So when they leave this room, the first thing they do is they go step and bleach to make sure that they don't have anything on them. They take their suits off. They bleach their suits. And their suits get destroyed that same day. They get incinerated. But before they do that, they have another person in that room inspecting them to make sure that there's no tears or punctures anywhere on their suits. They do that when they go in. They do that when they come out. If they find a tear or puncture, guess where you're going? You're going to quarantine and you're going to the hospital area. And you're going to stay there for 2 weeks. And they're going to monitor to make sure you don't have anything. Because this is stuff that they don't want to get out in the environment. We are very, very scared with the organisms that are in here. One of the ones that's also in here is one that we refer to as SARS. SARS is a cousin of COVID-19. We suspect COVID-19 was possibly manufactured based on SARS. And there's more and more evidence that's coming out of it, which explains why it's so easy to mutate and it's so easy to change. So we're still looking into that. But SARS is sudden acute respiratory syndrome. And SARS is another one that's a class 4. So we have to be careful with our class 4s. So here they are all laid out. We have BSL 1, 2, 3, and 4. What do we work with?
4.
Yeah, right. You said 4. That's right. Yeah, we work with BSL 1, and we have a few tubes that pop in there. Every now and then we'll have a tube that pops in there. We also, if you go to a hospital, they will work with BSL 3s. So a lot of times if you go to like a hospital that have a TB wing, they'll have special rooms that are negative pressure. They'll have signs on them, negative pressure. You have to have key cards to get inside. You're going to be doing your TB strains in there. The funniest thing, when I learned how to set up TBs, the person that trained me, hilarious old timer, he would smoke in the TB room because that way, nobody would catch him. And it was triple filtered, so nobody would smell it. And I was sitting with him, like, I'm working one of these hoods, and he's got like a cigarette hanging out, and he's like, here's how you do the patient sample. And he's like, smoking while he's doing TB. And this guy never turned positive. He was like so good at what he did, but it's like the old timer story. It's just like, good Lord, what we used to do. Okay, and then BSL 4s, never get near them, all right? These are the ones that if you ever want to read a fun book, read The Hot Zone. It's an older one now. But it's about the very first Ebola outbreak. And it's just an interesting story about how they went and tried to work with Ebola and stop the spread. So, did you read it? It's a good book, isn't it? Hot Zone.
Sorry, let's say you were to contract Ebola for some reason, right, randomly, and you go to your local hospital, you can't stay there. Where would they send them?
What they would do is they put you in isolation, they put you in a separate room right away, if that's what they suspected. And then what they would do is they would call around and find a facility that would take you. And usually it's going to be one that's going to be associated with like the military, because they're going to be the ones that usually have this class 4s. And then they will arrange to ship you to that facility. So if you can go towards CDC. Again, it just depends on if they have room for you or whichever military area has room for you with a class 4 lab that they can work on it. But the big thing is isolation. And that's where we run into problems. We actually had a very big scare in New York City. We had a patient who flew in from Africa, and while they were on the plane over the Atlantic, the lady that was sitting beside him, his friend, looked over at him and his eyes were bleeding. Can you imagine being on a plane, you look over and somebody's eyes are bleeding? Not a comforting feeling, right? So, of course, she calls the stewardess over, and the stewardess is trying not to panic, trying to stay calm. They tell the pilot they land in New York, and those people sat on the plane for over 10 hours while they tried to figure out what to do with this person, how to transport them, which facility to take them to. Luckily, it turns out that they had Marburg, which is a lower variant of the Ebola. So the person was treated. It took them several months, and they did recover. They managed to get them through. And luckily, because it was Marburg, it's not as easily transferred as Ebola is, which means all the people around him were not exposed, which is phenomenal. The worst thing is this patient flew knowing that they'd been exposed to Ebola, or that they've been exposed to Marburg. So that's the thing is if you're sick, please don't fly, because you're exposing everybody in that plane. Do you have a question?
Why do all the virus come from Africa? Why?
So they actually don't, but here's the deal. Where did people originate from? Where's the earliest ancestor we can trace back? Africa. So it turns out the reason a lot of the viruses and parasites are from Africa is they've been around us the longest. So they're able to adapt to us faster because they're so used to us. This area always had kind of lower populations. They haven't had time. Give them a couple million years and they're going to catch up. So that's the reason why a lot of them seem to come from there, is because they have more of a history with us. Also, here, how often are we exposed to bats? How often are we exposed to wildlife?
Not really.
Not really. I mean, we live in a bubble. If you live in areas like Africa, a lot of times you're living outdoors. You're more exposed to animals. And then you have more of a chance of a virus that they have jumping to you. All right? So that's the other reason, too. And also, where do we get our food from?
Stores.
The store. And everything's been taken care of. When I buy my meat, it's wrapped and it's clean and it's nice. If I'm living in a rural area, where do I get my meat from?
Lions or wherever you find.
Wherever you can, or you go hunt it. Right? So now I'm not getting it processed. It's not getting clean for me. I'm doing all of that, which means I'm getting exposed to the blood and the guts and everything that goes with that.
That's why you see on those shows where they check in customs, are you bringing food? Are you bringing those huge snails to eat?
Exactly. It's people, they'll check your food to see what you're bringing through. Okay, we're going to stop here, which means your quiz will go through the biosafety levels. Oh, isn't that nice?
Yeah.
I know. Kind of nice. I won't keep you late. Keep you more time. Okay. Do you guys want to start lab at 10:30 or 10:05? Since I went 1 minute ago. 10:35 it is.